So we're going to be handing over to Joe, who is in the studio there.
Good morning. You can hear me all right then?
Yep.
Yeah.
Here.
Happy International Microorganism Day, Ben. It's now.
Happy International Microorganism Day. I was checking the time zones all night. I think most of us are there now. I just have another quick look.
So we're waiting. No, the East Coast of America is now in International Microorganism Day. So it's just the West Coast that needs to catch up.
Come on, West Coast.
Wonderful. We're here. The day has arrived.
Yeah.
It is International Microorganism Day. I have my coffee. We have my co-host, Michael Sauer.
Okay. Well, then I'll bid you a happy Microorganism Day, International Microorganism Day. And I'll bid you farewell, and I'll watch in the shadows the rest of
Thanks a lot. I enjoyed the wine tour. I caught bits and pieces of it, but it was fun to see it.
Yeah, it was indeed. Well, thank you both, and enjoy the breakfast show.
Thanks very much. Right, Michael. I think we need to unmute you. We have you. How are you? Good morning.
How are you?
Yeah, not bad. I've had about 5.5 hours of sleep, which is better than none. But I wake up on International Microorganism Day of all days. So that is a fantastic place to start, isn't it? How are you?
I'm good. I just woke up. I had my coffee, and now I'm really excited about fungi.
Yeah, great. Because this year, you're my co-host for the breakfast show. Because last year, you were a guest of mine, and we talked about beer and yeast and baking and sourdough. And you had like a live sourdough culture on the table for us that we looked at. Now, this year, we're going to be looking through, lots of different things. And I'll just talk a little bit about how our breakfast show is going to commence, and then we can get on with it. But basically, we're going to be looking at our top 10 fungi. I mean, we might even have more than 10, but we're going to be working through a list of our favorite fungi for the first part. Then we have a couple of guests coming up. Our first guest will be Maria Martinova, who joins us from New York. So she's actually going to be joining us at nearly 2 a.m. there. But she's going to come talk to us about probiotics in yogurts. And also the Microbites platform, which is like an online kind of website, blog space platform of content for microbe lovers. Then we're just going to have a little recap of our favorite parts of the live stream so far. Celebrate the winners of the Microbe Art Contest. And then I think we've got a guest that you've lined up, Michael. Is it Paola Branduradi?
Exactly.
Yeah. And do you want to tell us a little bit about her background and what she's going to talk to us about?
Yes, she is from University of Milano Bicocca in northern Italy, but she actually is in Trieste now, traveling for a conference, so she will join from Trieste. She's a yeast specialist working on chemical production with yeast, and she knows a lot about really weird yeast, so it's very fun talking to her.
Well, that's going to be fun. I mean, yeast and fungi seem to be some of the weirdest microorganisms, at least in my experience so far. There seem to be lots of strange ones that do interesting and bizarre things. And yeah, I can't wait to discover more of them across the course of this show. But yeah, you joining us from Vienna, Michael?
Yes, right now, actually, I'm joining you from the western parts of Austria, but I am working in Vienna. So usually I'm in Vienna, working also at university there. But now I'm home at my parents' place. So when I watch out of my window, I see Switzerland, actually.
Oh, wow. So what town are you in now?
Oh, it's a tiny place called Mäder.
Okay. Is it anywhere near Salzburg?
No, it's much farther west.
Further west.
Essentially the westernmost point of Austria.
Yeah, okay. And how has Austria been treating you this past year and a half? I mean, it's been a very difficult year and a half, but have you been basically based only in Austria, unable to leave for the past few months, or did you manage to get some travel in over the summer?
Well, over the summer, I did some traveling, yes.
Yeah.
I went to the mountains, I went a little bit to the sea, but otherwise, that essentially was the first travel I did the last 1.5 years. Yeah.
Yeah, okay, wow. Yeah, just about...
home.
Yeah, well, Austria is not a bad country to be. One of the most beautiful places I've been. But I'm just going to recap to anyone who's joined us. What is International Microorganism Day? Well, it's a celebration of microbes for those who love microbes, whether you're a microbiologist, a student, or just someone who thinks they're cool. And the day was introduced by the Portuguese Society for Microbiology, and then the organization I work for and that Michael is involved in, FEMS, the Federation of European Microbiological Societies, have joined forces with the Portuguese Society to help try and internationalize it as much as possible. The date, 17th of September, which most people are now in, was chosen because it was the date that the first letter from Anthony van Leeuwenhoek reached the Royal Society, describing for the first time microorganisms that he'd observed with his homemade microscopes. So it's kind of, the human discovery of microorganisms, I suppose, that we celebrate today. Although they have been used by humans, right, throughout all of human history, pretty much going back to the point of agriculture, I think. People have found, beer brewing from thousands of years ago, fermented foods from thousands of years ago. They've obviously lived inside us and on us and, amongst our livestock and plants for the whole of human history. So they're super important. We're here to celebrate them. If you're waking up somewhere in Europe or across the world today, drinking your tea or coffee or even having your dinner, thank you for joining us on The Breakfast Show. I guess we shall just move into our first section because we've got just over 30 minutes before our first guest comes, which doesn't give us long per fungi. But we have a list of our top 10 fungi, and we're going to work through it today and discuss some of the finer aspects of these organisms. So, are you ready, Michael? Are you ready for our top 10 fungi? Okay. So we put this list together, and I don't think we fully decided which were our top 10. So we might have a bit of argumentation or debate across the course of the next half hour about which should make it into the list. But for me, the first one on the list that I think is worth a mention is the Cordyceps fungus. Now, are you familiar with this fungus?
Yes, a little bit.
A little bit.
It's so weird that I also looked into that one.
Yeah, because it is one of the weirdest fungi that I've ever read about. So if you haven't heard about Cordyceps, let me just explain a tiny little bit about it. So this is a species of fungi that basically infects insect species, and it starts to grow inside the insect and replace the insect tissue with fungus tissue and hyphae and so on. But it doesn't just sort of rot the microorganism, it actually takes control of its brain and nervous system. And in a little minute, I'll have a couple of images for you, but what it essentially does is control the insect so that it starts to do strange things like walk up a tree or a plant and bite down into the top of the plant. And then once the insect's dead, the Cordyceps fungus fully replaces the internal size of the flesh and then bursts out through the insect body with its fruiting bodies. So it's kind of a zombification fungus that also manages to control and then digest the organism. Do you have any fun facts about Cordyceps, Michael, before I show us a small video?
Well, a fun fact that I recently came across is how wonderful evolution works, because exactly as you said, so it takes control, it makes the end go up to the tree, what the end naturally would never ever do. And then it bites there. And why does it do so? Of course, the fungus wants, let's say, to spread the spores as high as possible to get, a large area. And usually they are from tropical areas. So that's where they evolved. And there the ants bite into leaves. But they spread actually in areas where more colder weather is. And there it's not good to be at the leaf because the leaf might fall down at the wrong time. And so actually it would be better to bite in a little, woody part of the tree. And actually it evolved that way. So there are cordyceps which make the ant go up the tree, but bite into woody parts and not into the leaf. And that's completely weird because that means that this fungus can really control not only how the ants behaves, but also where the ants is, so depending on the climate.
And they are found everywhere or in lots of different environments, right?
Nowadays in lots of different environments. I think in Europe they are not really present.
So here we have a picture of a Cordyceps fungi bursting out of a dead wasp. So you can see these fruiting bodies. I mean, they're like little mushrooms, really, for the Cordyceps coming out of the body of the wasp. But again, this wasp, as you say, has bitten down into a woody stem. And I guess this is to place it somewhere, you know, in a high exposed position so its spores can then float out over the jungle or the forest and infect other insects. But there's a few other weird facts about this fungus. And do you know about the role it plays in Chinese medicine?
Yes.
Yeah.
Well, the point is, it's such a weird thing, and that it's allegedly it has a lot of different roles, medical applications, and so it's extremely expensive, to be honest. So if you want to buy certain kinds of the fungus dried and milled, it's quite expensive.
Yeah, and in fact, in some places in Bhutan, villages in the mountains, one of their main sources of income is collecting these Cordyceps bodies, which look a little bit like this.
Yeah.
And then selling them on to traders. And it's their biggest source of revenue in some parts of Bhutan. This is a paper, actually, from our journal, Fem's Microbiology Letters, that looks into the use of Cordyceps fungus in Chinese medicine. But one of the things they highlight is that, yeah, like you say, they're so expensive and worth money that people have actually begun faking the Cordyceps fruiting bodies. So of these 4 here, example C is actually a fake one. So these are real caterpillars with a fruiting body bursting out due to the Cordyceps fungus. But this version here is simply a caterpillar and a grass stem that has been glued together to fake a Cordyceps fruiting body to make some money. But you can't really tell, can you? They're very similar. But this paper I dug out from the archive from 2013 basically looks into, they're barcoding the DNA, the little caterpillar funguses here, and they found out that some of these are fake. So not all the fungus that you will buy in the Chinese medicine market will be the true Cordyceps. So I think that's a great one to begin with. I think the second one on our list is probably the favorite, maybe of you, Michael, but at least of many microbiologists. So that one is the famous Saccharomyces Cerevisiae.
Yes, exactly. So that's the brewer's yeast or baker's yeast. And the point is, first of all, I think it's good to talk about it after the creepy one, because that's the one that gives us lots of good things. So it gives us beer, it gives us wine, it gives us bread, and it's very useful. And actually, that's what microbiologists tend to call the conventional yeast, because when they talk about other yeasts than baker's yeast, they talk about unconventional yeast. I think it's kind of funny that only one is conventional, but that's why we know a lot about it.
So we've got one conventional yeast, and then everything else is unconventional.
Is unconventional, exactly.
Fair enough. And then we have a lot to thank, right? I mean, so we have beer, wine, which has had a vineyard tour. So there would be no wine without Saccharomyces Cerevisiae. Bread, it's involved in some way, yeah. I mean, I'm British, so...
Pizza.
Sorry, say that again?
Lots of cakes and also pizza.
Pizza. Cake, pizza, yeah, okay. All of the delicious things, it seems, are thanked. We can thank this yeast for. And I was going to say, I'm British, so I love Marmite on my toast in the morning, which is made from the boiled up yeast bodies of the brewing process. So I think they boiled up all of the yeast at the end of the brewing process. They dehydrate it into a delicious, sticky black mess, full of salt and vitamin B12 and tons of other good nutrients. Oops. So here we have a little video again of Saccharomyces cerevisiae. So you can see, you know, it's fairly simple looking, isn't it, under a microscope? It's just a little budded yeast cell, which forms these little sort of colonies almost of grouped cells. But here we can see the budding process, right? So it doesn't reproduce like our cells or bacteria. It does a slightly different process. So I wondered if you could tell me a little bit about the budding reproductive cycle.
Yeah, that is extremely interesting. The point is the budding is an asymmetrical process. So it's not like bacteria usually divide in the middle and then you have two equal parts. And this is also quite philosophic because bacteria in this sense live eternally because, you know, every part can divide again. But in yeast, this thing already became asymmetrical. So what you can see here is that a little part is budding from, let's say, the bigger part. So the little part is the young part, we call it the daughter cell, while the bigger part is the mother cell. And so it's really not only asymmetrical in size, but also like the molecules are divided. Let's say all the new ones are going to the daughter and the older ones are sticking to the mother. And that means already that here the eternal life ends, because the mother can only divide a certain number of times and then it dies. So yeasts do not live eternally. They have daughters and they go on, of course, but the mother is doomed at one point because it accumulates, let's say, the old stuff.
Okay, so like a well-meaning and I guess motherly parent, it gives the best of what it can to the new cell.
That's exactly what it does. That's exactly what it does.
So, I mean, we had a brewery tour last night, and they were saying that, they probably got out about 20 generations of yeast reproduction from their yeast before it started to perhaps decay a little bit and not be quite as snappy as it was in the past. So, I mean, that doesn't make too much sense to me at the moment, because doesn't this mean that like the yeast will reproduce for 20 generations, but then suddenly it can't reproduce anymore?
Well, the point is you always have the daughter, right?
Okay, so it's just the original cell.
The mother, exactly. The original cell at one point eventually dies, but the daughter then becomes a mother herself and will start to bud again herself. And so you still reproduce, you still get more and more and more. So one can do it like 30 times. So you get, unlimited amounts of yeast cells, so to say.
Yeah.
But if you look at the individual, then there's a difference between bacteria and these yeast cells. So, and it's interesting because it's like growing from the really simple ones to the really complicated ones. Humans, at one point, age becomes a thing. And yeast cells age, and you can see aging, and you can study aging with yeast cells.
Because actually, I think you're an editor for one of our journals in Microbiology Letters, but we have a corresponding journal called Fem's Yeast Research, right, which focuses just on yeast and other similar fungi. But they had a thematic issue on yeast death and cell aging, and I guess we can use it as a model organism for studying human aging in some ways, isn't that right?
Exactly. Yeah. Because some of the mechanisms that happen for yeast cells are very similar to the ones that happen to us.
So hopefully, not only can yeast provide us with all the wonderful foods and drinks that we love, but maybe it will unlock the key to immortality, and it will make us live forever.
Exactly.
I think number 3 on our list is another yeast, which I didn't know about. So you've added this one. So that's Saccharomyces Pasteuranius.
Yeah, the point is when we look at lager beers, they are actually not produced by Cerevisiae, but they are produced by another yeast. And the interesting story behind that is that this yeast is kind of cold tolerant. So lager beer is brewed as a lower temperature than ales, for example. But the yeast has to be efficient. So selection actually led to a hybrid cell. So that's also kind of a zombie existence. So what happened is that 2 yeast cells fused and, let's say, built a new entity in the end. And what was known very early when we studied these lager yeast cells is that one part was Cerevisiae. So that's the part that brings in the good fermentation and the aromas and all this stuff. But Cerevisiae is not cold tolerant. And so genetically, what we could figure out is that there must be another part, but we didn't find it. And many people were looking long time until it was found actually the first time in Patagonia.
Okay, and that's the first time it was found, but it had been used for...
The first time it found that it was a big thing. So breweries were using that yeast and so on. So it's absolutely fantastic. And so there was a lot of debate because, of course, the yeast itself was selected in Europe. And so the question was, how would the yeast from Patagonia come to Europe? So there were a lot of weird stories about this. The point is, then this yeast was found also in North America. Then this yeast was found also in Tibet. So it's found in many places on the planet. But interestingly, so far it has not been found in Europe. So we still don't know exactly how this all came into happen. But it's really like a criminal story. And if you read the different publications from different groups at different parts of the world, it's really fun to read. Because of course, everyone wants that his yeast is the ancestor of all our lager yeast strains.
Yeah. So found in Patagonia, but it is used in beer brewing.
Worldwide.
Now it is. Okay, but it hasn't. That's a very strange crisscross of the microbes across the world, I guess.
Exactly.
Yeah.
There seems a lot of traveling involved.
There definitely does. And I always wonder how these microbes cross the world because, individually they can't really move around that far. But I guess they hitchhike maybe on other animals or...
Oh, they hitchhike. For example, particularly talking about baker's yeast, we know that they hitchhike in bees and wasps. So actually, one of the questions is, how do they survive, for example, wintertime? Because in Europe, when everything freezes, how can they survive? And how would they come back to fruits and flowers? And what we figured out is that when these bees and wasps go to flowers and fruits, they ingest also the yeast, and the yeast survives in the bee. And when it goes back to the hive and stays there over winter, actually also the yeast stays. And then, like with a helicopter, they go back in springtime to all the flowers and fruits again.
Wow. Funky way of traveling.
Yes. What is very interesting is that fermentation, for example, has a connection there, because a lot of these insects are attracted also by fermentation products. In this case, it's not like for humans the alcohol itself, but more some like aromatic side products. But the insects are attracted by it, and the yeasts do it. So essentially, the yeasts kind of make fruit and flowers attractive for the insects. So they tell them where to go, where they want to be, and where they want to mingle with other yeasts.
And how do they do this? Is it a chemical signal or something, or
yes,
are they
just
signal?
It's like smelling. You can imagine, really, it smells good for the animal.
Okay.
It's like the beer, the fresh beer, the fresh bread. It smells very good for us. So that's exactly what also the bee has. And so the bee goes exactly there. And then the yeast is happy because it arrives exactly at the point where it wants to be.
Very nice. I'm just realizing we're only on 3 and we have 17 more minutes before our next guest comes. So I guess we should move on to the next one on our list, which I think I put down here. And this one is Acrimonium. Which, if I just get up this small video, I put it on this list because it is a useful one. It basically gives us the Cephalosporins, a beta-lactam antibiotic. You said to me that this might be a bit boring, but I think it's cool because in general, it provides us with, such a useful group of antibiotics. I think in terms of the biology, it's a fairly mundane fungi, right? It has spores and hyphae, and there's nothing super crazy about the way it lives its life. But that's okay. It lives an ordinary but useful life. So here you have just a few shots. So I guess it lives in blackberries or on fruits or, on different kinds of plants and so on. And you can see here the spores, right? Which are how it reproduces. It produces these small spores and then they go off and find new locations and then they eventually grow into a fully grown Acrimonium. But I want to just move us on to the next slide because we can have a little look at this antibiotic. So here we go. Here we have the Cephalosporins. So I guess it's good to talk about this in comparison to penicillin, right? So all of these antibiotics have this strange nitrogen, carbon, carbon, carbon square with a double bonded oxygen, which is the beta-lactam ring. But I think, if I remember from my studies, what makes these antibiotics a little bit more durable for treating disease than the penicillium or penicillin, which we're used to, is it has this extra ring here with a sulfur atom. And this makes this molecule much harder for sort of beta-lactamases, which are enzymes that break down antibiotics, it makes it much harder for bacteria to be able to break this molecule down, because this sulfur gets in the way and this ring is a bit different to what it is in penicillin. So it provides us with a useful alternative, and I think that's really cool, because as we know, antibiotic resistance or antimicrobial resistance is a huge problem, and it's little chemical changes, like small little bits of sulfur that you add into molecules, which might make these molecules last a bit longer. So for me, that's why it's on the list. I guess as a biologist, perhaps you say it's not the most interesting.
Oh no!
Or at least.
No. Poor Acrimonium. I didn't want to say it's boring in itself. It was just that when we compare it to penicillium, the point is also penicillium gives us the antibiotics, and penicillium is the historic one. And penicillium gives us wonderful cheeses and other things.
I mean, it is number 5 on our list.
Exactly. That's why I thought, okay, let's talk about penicillium to save time, but not to avoid Acrimonium, which is, as you say, the point is what we need is diversity, not only in our societies, but what we need is diversity also in antibiotics. And that's exactly what, the more fungi we have, the better it is.
Yeah. And of course, the first kind of true antibiotic came from penicillin, discovered by Alexander Fleming. Florey and Chain, and I'm sure many other scientists at the William Dunn School of Pathology helped turn this into a product we could mass produce during the Second World War. I actually had the honor of being a laboratory intern at the William Dunn School of Pathology at Oxford University for 3 summers, working in the same building that they did all this process in, which was amazing. And, you really feel like you're walking in the footsteps of giants. And yeah, I mean, you find these molecules, you might find them produced by fungi or other microorganisms, but then turning it into a product that is safe and effective and also mass producible is the next challenge, right? So, we need scientists looking for molecules like this, but then we also need to work out if they're going to be therapeutically useful. So hopefully fungi can provide us with more examples of this. And here we go. Here's a nice little example of some Acrimonium growing on some agar. You can see the nice little hyphae here. But penicillin gives us the original beta-lactam antibiotic, but it also gives us blue cheese. And in fact, it's Penicillium roquefort, which John Warhol, back last night, was saying should be the official microbe of the town of Roquefort, where they make the blue cheese itself. And I think that's a great idea. So if you're listening from France and you're near that town, please petition your local legislators to make Penicillium roqueforti your official microbe. But there's another one, isn't there? Penicillium camemberti, which I think the name gives it away, helps us make brie and camembert. So from saving lives to giving us cheese, we really have to thank penicillium. Right, okay, we've got 10 more minutes.
Things, right?
Say that again?
They can do a lot of things.
Yeah, they can. It's amazing. I mean, I had cheese for breakfast, so it's even powering me through International Microorganism Day. Yeah, we've got 10 minutes to do 5 microorganisms, so I guess we better get our move on. So the next one is, it's let me just get this ready here. It's, Malassezia. So do you know about this one, Michael?
Not too much, actually. You brought it up, and then I had to look it up. And the point is also there's a lot of philosophy behind. The point is, this is a fungus that grows on our skins. And it's there for all of us, essentially. Most people have it. But then the question is, what happens?
Yeah, exactly.
Exactly, the film.
And it grows on our skin, and usually it's beneficial. And much like yeast, it does budding as its process for reproduction, and we can see a nice image here under some scanning electron microscope. But when this fungus gets out of balance, I guess, with its neighbors, you can see a little more images here, it can cause some problems with your skin. So one of the things it can do is it can start to change the pigmentation of your skin. And if you have a weakened immune system or, some kind of perhaps nutritional deficiency, it can cause hypopigmentation, which is a reduction in the pigmentation of your skin, or hyperpigmentation, an increase in the pigmentation. So I just think that's a very strange and local fungus, which can have a huge effect on people's complexion and skin. So it's a very like human-centric way of looking at it, but it is quite an amazing thing that it can do. So that's why it's on my list, because it can, completely change the color of your skin. And it's normally living on everybody, right? So it's something that we all have somewhere on our skin, in our hair and so on. You can see a little bit more of the structure here. It has a thick capsular layer and thick cell wall. So it's pretty well defended against things around it. Next one is Aspergillus, which is quite a famous one, actually.
Yeah, and that's one of my favorites.
Do you want to give us a little introduction while I get this image ready?
Yeah, get an image ready, because
here we go. This is what we like to see.
Exactly, that's very beautiful. Yeah, Aspergillus can also do a lot of things, but one of the things that it's really useful for is for citric acid production. So when you drink a lemonade, or when you cook your jams or something like this, and you add citric acid, that's actually usually not extracted from lemons anymore, but produced with an Aspergillus strain. And this fungus is extremely efficient in eating actually sugar and converting it into citric acid. And that's what hardly anybody knows.
Wow, okay. So, I mean, just to put it in context for anyone listening, citric acid is that kind of tang-fastic flavor that you get on sweets. So if you ever have a very sour, tangy sweet, it's usually been dusted in citric acid. But our bodies use it, right, in their metabolism in many different ways.
Everywhere. It's absolutely everywhere.
Yeah. And so they turn sugar into citric acid. Okay, that's a cool fact I didn't know. And it's a mold you might encounter, I guess, if you leave a tomato to go moldy in your kitchen and you see it go white and furry. That could be Aspergillus. And in fact, here we have a close-up, and then I guess these are the fruiting bodies ready to spore and then infect the rest of your tomatoes. And do you work with Aspergillus at all?
Yes, we do work with Aspergillus in our lab.
Yeah,
and we're actually looking into it, and the point is, they are always so why we as humans, we always try to explain something with our ideas, and most of the time these questions might be even stupid because there is no really a why. But the question is, why do they produce so much citric acid? It seems completely futile, because they can really eat the sugar and convert 95% into citric acid and grow only very little with it. So the question is, why do they do that?
So that's an unknown. So if you're a young budding scientist looking for problems, there's one to investigate.
Exactly.
So another favorite fact of Aspergillum is, and we might see this later today, because after this we have a session on traditional Japanese beverages, including a tour of a sake factory. But the koji mold used in Japan to ferment soybeans, but also to make sake, is actually an Aspergillus species, Aspergillus oryzae. So it's also used in making sake. So we have another fantastic use for this fungus. I think
They produce a lot of enzymes, and so they're very useful. Like sake is produced from rice, right?
Yeah.
And rice is very starchy, but the yeast that actually produce the alcohol wants sugar, doesn't want starch. And so the fungus actually can degrade the starch into sugar that the yeast uses.
Fantastic. And actually it gets its name from this structure, I think, called the aspergillum. Is that right? Yeah, which is the fruiting body. You can see all the spores here in this close-up. Okay, that's number 7 on our list. We have a few to pick from. Do you want to pick a top 3 or the next one from this list? And we'll try and get through the last 3 in the next 5 minutes.
Yes, let me open the list myself. Yeah, why not go directly to the next one, to Yarovia lipolytica?
Yeah, okay. I don't have a video of this, but if you could give us a little introduction, that would be great.
Yes, because Yarovia, we talked about like when yeast gets not eternally living anymore. So there are a lot of these steps how organisms develop. And Yarovia is kind of a thing. Yeah, there you see it. You see actually that the middle picture is fantastic.
Ooh, let's go back. This one?
The one below, this one.
This one.
Is good, yes. What you can see here is that the cell can grow as a yeast, like you saw it before from baker's yeast. So you have these little cells and they are budding and they look exactly like the yeast that we saw. But then you see this long hyphae also, right?
Yeah, I can see.
Much more like the Acrimonium or like the Aspergillus. And so Yarovia is kind of an in-between on the way between yeast and filamentous fungi. So it can do both. Depending on the conditions, it can completely grow as yeast, or it can completely grow as hyphae, or it can do both at the same time.
Wow. Okay.
Really cool to study, because also we don't understand how exactly that's regulated, and so that's a good thing to study it.
Okay, so at some point, some genes are getting switched on and off in different environments, and we don't quite know the process.
Exactly, and the hyphae cling to something. The other really fun fact about this is lipolytica already tells you a little bit. So it can actually degrade fats and oils and grow on them, what is special. Many things cannot.
So it might have a role to play in clearing up oil spills, perhaps, or something, although it's not aquatic, but it can definitely eat lipids.
Yes, it can eat lipids, which is biotechnologically interesting. And for example, in our lab, we are working with a strain that has been isolated from the tank of an airplane.
Wow, okay. So it's living in there somehow.
Exactly, it's living in there, and it can even degrade something like kerosene.
Whoa. So it can survive off jet plane fuel.
Yes.
And it can pretend to be a yeast type thing or a hyphae type thing.
Exactly.
Okay, that's definitely number 8. So we've got 3 minutes to do 2 fungi. So any on this list that you really, really want to get to?
So let me look. Wait a second. Well, later on we have Zigo, so that we can. So let's go to Trichoderma very briefly.
Which one? This one.
Number 10.
Yeah. Let's see if we can find us a nice image.
Because again, fungi give us a lot of molecules like antibiotics, but also a lot of enzymes. And in our quest to produce things, for example, from woody biomass, we need enzymes to degrade wood, for example.
Yeah.
And this one is one of the best producers of enzymes degrading wood. And it has been found actually in the Second World War from American soldiers on the Solomon Islands, because what they realized is that their canvas and their paper was falling apart under their hands. There was something growing on it and degrading it. And it was actually this fungus. And then we figured out that it has fantastic enzymes. And with these enzymes, we can degrade a lot of things in our environment, and we can use it. And so from looking at some damage somewhere, we could get a strain that is now one of the worldwide #1 producers for enzymes.
Whoa, okay. So from degrading soldiers' papers in World War II, it's now an industrial giant.
Yeah.
So here we have a little image of it growing under the microscope, but then this is a more macroscopic sort of big spore colony thing with fruiting bodies as well. Okay, we've got 1 minute, 1 fungi, 1 fungus. What's our 10th fungus on this list?
Let's go to Exophiala dermatidis.
Yeah.
It's black yeast, and that's a cool one because it's so sturdy. It grows extremely slowly, but it grows everywhere. So there's hardly any condition that it cannot grow in. It's black, it's cool, it really looks nice, I think. And it can grow, for example, in your dishwasher. So you wash your dishes with 90 degrees, you have very, very aggressive dishwashing agents, but this yeast can survive that, and it will not die, it will grow. It can grow on glaciers, it can grow everywhere. And that makes it very interesting for us, because really life is everywhere.
Whoa. So it can grow in dishwashers at 90 degrees. It can grow in glaciers at minus whatever.
Yes.
Okay, that is a hardy fungi.
Happy. The point is that's a problem, of course, because you don't want a yeast in your dishwasher. So, but for the yeast, obviously it's cool. And
I mean, that's a huge range of environments that it's happy in. Exactly. And it's this, so does it make these black colonies, these black kind of groups?
Exactly.
Whoa.
Aren't they cool?
They are really cool. I didn't know about this one, so I thank you for adding it to the list. Unfortunately, we've run out of time. I mean, we could go on talking about fungi, it seems, for hours. And we've just managed to cover 10 in the last half an hour. We have our next guest arrived in the call. So I'll just invite her and stop sharing my screen. And we'll just check that all of her equipment is working. But yeah, that was fantastic. I mean, we could have gone on for hours, I believe. We really had to rush through just a very